Welcome friends, myself Akash Bhise, first year MSc Agriculture, Postgraduate Institute Akola, discipline plant pathology. So friends, let's see some information about seed-borne fungus of soybean. Soybean is one of the most important oilseed crop in the world and also a good source of protein and fiber. Soybean contains 40% protein and 20% oil. Soybean not used as dal purpose due to presence of harmful substance lipoxidase. Botanical name of soybean is Glycine max. Family Fabaceae. Origin China. It is self-pollinated crop. In terms of production, USA rank first in the world. In respect to India, Madhya Pradesh and Maharashtra are the major soybean producer states. Friends, there are lots of fungal species that affect quality as well as quantity of produce. So provide the primary inoculum for the diverse soybean disease in the soybean, while movement of these fungi through infected seed brings an important challenge to the global soybean production, as seed may travel across the world. The fungal species are identified by the two ways. First one is laboratory method, while another is visual symptoms. Laboratory method includes standard blotter paper method, 2,4-D blotter method, deep freeze blotter method, and agar plate method. While external symptoms shown by an plant due to the attack of fungus comes under visual symptoms. Sometimes it's difficult to identify the fungus by external symptoms. Some common seed-borne fungus of soybean include Caletotrichum truncatum, Fusarium oxysporum, Alternaria alternata, Carularia lunata, Chitomium globosum, Aspergillus flavus, Aspergillus niger, Penicillium, Rhizopus, and Cladosporium. Caletotrichum truncatum. It is plant pathogen cause anthracnose to soybean. We can see it shows symptoms on leaf, pod, seed, and seedling, while fungal spores are observable under compound microscope. Fungus produce crowded black aceroli on infected tissue. Symptoms: when infected plants are planted, pre-emergence and post-emergence damping up occur. Sunken and dark brown lesions develop on the cotyledons of seedlings. Seedling lesions may expand to the stem and kill young plants. Plants may infected at any stage of the development. Fusarium oxysporum. This pathogen causes Fusarium wilt in soybean. Fusarium wilt is one of the major limiting factors to a soybean production. Infection can occur at any growth stage, but more often occur at early vegetative and reproductive stage. Fusarium oxysporum is an ascomycete fungus. The pathogen can observable under compound microscope. Fusarium wilt often occur in irregular patches in the field. Symptoms are often observed in hot and dry conditions when plants struggle to take up water. Earlier symptoms include intervenular chlorosis, premature defoliation, and wilting. The root often decay with brown to black epidermal tissues and reddish to brown vascular discoloration extending into the stem. Alternaria alternata. This pathogen causes alternaria leaf spot of the soybean. Alternaria leaf spot occasionally appear in seedling stage but generally attacks leaves and pods of the plants. Diseased lesions are round or restricted by major veins or merge with another lesions. Infected leaves eventually dries out and fall. Spores are look like this actually when seen under compound microscope. Carularia lunata. This fungi cause cotyledon spot of soybean, produce and necrotic lesions on soybean. Infected seed shows slight discoloration similar to Caletotrichum species. Seed coat takes on an reddish tint to underlying cotyledonary tissue become light brown. This disease is minor importance. Spores actually look like this when seen under compound microscope. Aspergillus niger. This pathogen causes black mold. Species within Aspergillus genus are often fast growing and can correlate within a few days after germination. Aspergillus niger is a filamentous fungus capable of growing in a variety of environments due to its versatile metabolism. It is also produce mycotoxins called acratoxins when observed under compound microscope, shown in black filamentous fungus. Aspergillus flavus. It is saprotrophic and pathogenic fungus with cosmopolitic distribution. Cause post-harvest rot developed during harvest and storage. Aspergillus flavus infection can occur while hosts are still in the field. that is pre-harvest, but does not show symptoms oftenly until post-harvest storage. Aspergillus flavus colonies are commonly powdery masses of yellowish-green spores upper surface of seed. Hyphal growth usually occurs by thread-like branching and produce mycelia, hyphae are septate and hyaline. Other seed-borne diseases like Cercospora kikuchi. This pathogen causes Cercospora blight. Dark lesions may not always be present on pod, but pink to dark purple discoloration occur on the seed coat. Cercospora sogina. This causes frog eye in soybean, which shows circular to oval lesions that are red-brown to black on pod, while reddish-brown lesions often seen on end of the seed. Phomopsis species. This pathogen causes Phomopsis seed decay, which shows black fungal specks, that is pycnidia, on infected tissue on plants, while seeds are cracked, shriveled, and chalky white appearance. Seed treatment. To control the seed-borne fungus, the seed treatment is important. The correct fungicide is critical for effective disease control. Fungicide used as seed protectant, that is, contact fungicide, are effective only on the seed surface, while if we use systematic fungicide that are absorbed by germinating seed, it kill or inhibit the seed-borne pathogen. Contact fungicide have usually shorter residual activity than systematic fungicide. Contact fungicide are used for soybean seed treatment include Captan, Fludeoxonil, PCNB, and Thyram. Systematic fungicide used for soybean seed treatment are azoxystrobin, carboxyn, mefenoxam, metalaxyl, trifloxystrobin, thiabendazole. Thank you all of you for visiting my channel.